The overall goal of this protocol is to obtain, visualize, and quantify the 3D chemical maps obtained by energy-filtered electron tomography.
This method can help answer key questions in material science, chemistry, and catalysis fields, such as how the components of a given material are mixed at nanometric scale. The main advantage of this technique is that it provides information concerning the chemical distribution in the bulk and at the surface of the sample. Energy-filtered imaging can separate chemical elements that are very difficult to distinguish using other imaging techniques. The experimental protocol described here shows the means of obtaining 3D chemical maps, but also it tries to provide ideas to quantify some of the 3D chemical parameters. The 3D chemical maps can be only generated for samples that are not beam sensitive, since the recording of filtered images requires long exposure times to a relatively intense electron beam.
To begin, prepare a sample such as silica alumina or titania alumina as described in the accompanying text protocol. Next, use an electron microscope to record energy-filtered tilt series. These images will be used to calculate each chemical map of the search chemical element. Open the 3 filters.